So again, this is going to be for aerobic respiration. So let's just start off with just titling this thing. Stay really up close to the top though, because we're going to end up using like this whole paper. If I start to go off the paper, let me know, because I'm going to be trying to go fast here. So glucose plus oxygen is going to go to CO2 plus water plus, and again, we always said ATP, but really what's going on is we are releasing free energy that is then used to make ATP, and I'm already off. It's just what was already on the board. So this is the overall process. Now, the pathway to get there is a long and twisting one. So we're going to break down each step within aerobic respiration and make this as simple as possible. Okay, there's 4 primary pathways in aerobic respiration. What are they? Glycolysis, and then the Krebs cycle, also known as the citric acid cycle, also known as the TCA cycle. There's 3 different words for it. Ridiculous. Okay, and then where? Transport chain, and then oxidative phosphorylation. Okay. So we are going to start off in glycolysis, and what I'm going to do is just kind of shade in the box. I'm going to try to color code this whole thing. So like, I'm just going to outline this box in blue to show that this is where all of the glycolysis stuff is going to be that we talked about. Because once we start moving all these arrows around, it's going to be a lot easier if we have a color code to know like where stuff came from and where it actually went to. All right, so we're starting off with glycolysis up here with an entering glucose molecule. And again, it doesn't have to be glucose. It could be any large biological substance that's found in our food. So instead of glucose, well, along with glucose, I'm just going to put in parentheses here, food. I mean, it could be sucrose. It could be triglycerides. It doesn't matter what it is. At some point, any of the nutrients that you consume that contain energy will enter this pathway somewhere along the line. Okay. So we start off with glycolysis, and the overall idea of glycolysis is going to be the oxidation of glucose. And again, we keep saying of glucose, but just know it could be many different things. Oxidation of glucose into how many pyruvates? 2. 2 pyruvate molecules. Okay. And then along the way, some things happen. So you don't need to know all the 10 steps of glycolysis, because holy cow, who has time for that? We don't. So the first thing that we're going to see in glycolysis is there's going to be an energy investment and then an energy payoff. 2 ATPs have to go into this system. The reason behind it does not matter. And then at the end, we end up getting 4 ATPs out. So we are going to have a net gain of 2 ATPs. And that happens up here. Here's our ATP. Every time there's ATP, I'm just going to shade it in as orange to kind of draw our attention to it. There are 3 different places that we get ATP in aerobic respiration. We get some in glycolysis right here. We get some in the Krebs cycle, and then we'll get a lot in the electron transport chain via oxidative phosphorylation. Okay. Then this glucose is going to start moving down the pathway of glycolysis. It's a 10-step process, and you don't need to know all 10 steps. But at the end of this thing, two things are going to occur. The next thing that we see, so I'm going to be like putting a 1 here to say, okay, this is where number 1 happened. Okay. Step 2, the next major thing that we see, because this way we can basically break glycolysis down into 3 major steps. The next thing is we're going to make some NADH. So we are going to produce 2 NADHs. This occurs through oxidizing. It's not glucose anymore. It's one of those, intermediates of glucose as we move down this chain. But we're going to oxidize what is left of this glucose, which means we're going to take electrons off of it and put it onto NAD+ to make NADH. And NADH is an electron-carrying molecule. It carries free electrons, it's very high energy, and those electrons are going to be transported to the electron transport chain. So we produce 2 NADHs, and those are going to enter. I am never going to write out electron transport chain. Starting now, forever and always, I will abbreviate it ETC. If you see ETC, that means electron transport chain. Enters the ETC. So let's shade in that arrow to show it going down here. So here's our NADH. It's leaving glycolysis. And it's going to go all the way down if you follow this thing down. I don't know what kind of movie The Polar Express is, but it does not sound like a train movie. I'm just saying. Is that what that is?
It's a dinosaur.
Come on, that is not Polar Express. Come on. Alright, anyway. So, we have NADH formed, leaves the glycolysis reaction, and it's going to go into the electron transport chain. That is one of the places that electrons enter from, is through glycolysis. And then the last step, this is step 10 in the entire process of glycolysis, but for us it's just going to be step 3. So 3 up there. We are going to make 2 pyruvates. So 2 pyruvates are produced. Pyruvate is a 3-carbon, it's a 3-carbon molecule. So basically glucose is a 6-carbon molecule. Pyruvate is just glucose cut in half with some hydroxyl groups missing and some other stuff. Okay. We don't need to know the overall structure of it. Just know that glucose is oxidized into pyruvate. This all happens outside of the mitochondria. At this point, once we have these pyruvates, there is a, I'm going to use scare quotes, a decision to be made. And that decision is dependent on whether or not there is oxygen. If there is not oxygen, what pathway does this pyruvate enter? Fermentation, which is what you're going to review using that assignment after tonight. So that'll get your fermentation review. If there is oxygen, though, then we are going to start what's called the link reaction, which I don't care that you know that, but there's this weird space in between the Krebs cycle and glycolysis where we have to take this pyruvate and get it ready for the Krebs cycle. I'm going to, your book includes it as part of the Krebs cycle. Just know that it's technically part of like this kind of in-between reaction. So the next thing that we have to do is we have to get pyruvate into the mitochondria. How does that happen? What process has to occur? How does pyruvate get into the mitochondria? What kind of transport? Active transport. So we're going to see active transport here. Does active transport require ATP? Heck yes, it does. Okay. I'm going to go ahead and outline our pyruvate in blue because this is the end product of the glycolysis pathway. Okay. So active transport is then used to get this pyruvate into the mitochondria. I'm now switching to red because everything now is going to take us into the Krebs cycle. Okay. Our Krebs cycle or citric acid cycle box is way down here. So I'm going to be coming down here now. All right, step 1. Pyruvate enters the mitochondria via active transport. Sorry about my handwriting. I'm trying. That's really not helpful. You okay?
Yeah.
Okay. Then, 3 things have to occur to pyruvate in order for it to be used in the Krebs cycle. Do you remember what they are? Yeah. 3 things. CO2 is released because we get rid of the carboxyl group. So number 2. So the first, this is the first part of the link reaction, is the removal of the carboxyl group. And it's released as CO2. I'll just say as CO2, save space. So removal of the carboxyl group is CO2. Okay. That would occur right here. You can see our CO2 spiraling off of the reaction. So I'm going to outline our pyruvate here. I'm going to draw in our arrow. Here goes that CO2. And that is step 2. Okay, good. What else needs to happen? It's right here. Yeah, so we're going to make some more NADH, and this occurs whenever we oxidize this pyruvate. Remember, oxidation is the removal of electrons. So electrons are going to leave the pyruvate and be used to reduce. So this is a redox reaction. If you oxidize something, you can use those electrons to reduce something else. So when something is reduced, it's gained electrons. So electrons are going to be stripped out of the pyruvate and put onto the NAD plus to make NADH, and then the NADH is going to transport those electrons to the electron transport chain. We're stripping out electrons. We're removing energy from the pyruvate. This entire process is just harvesting energy out of glucose. That's all we're doing. We're just stripping it out a couple electrons at a time. Okay. So on number 3, then I'll say oxidation of pyruvate generates NADH. So now we're taking more electrons to the electron transport chain, and that enters the ETC as well. ETC. And then we can shade in that arrow. So here we have some more NADH headed into the electron transport chain. Anytime you see NADH entering a pathway, it's taken electrons away from somewhere else and it's transporting them to another place. Okay. Step 3. I guess that was 3. Step 4. This is the last thing that has to happen before it can enter the Krebs cycle. So the Krebs cycle, or the citric acid cycle, starts off with a molecule called acetyl coenzyme A. The pyruvate at this point is only acetyl. It's an acetate. It doesn't have the coenzyme A yet. So the fourth thing that needs to happen is we have to add coenzyme A. So this would be number 4. Coenzyme A is added, forming acetyl coenzyme A. Alright, what makes this confusing is all of the weird words. So oxidation, pyruvate, NADH. So oxidation just means removing electrons. Pyruvate is broken down glucose. NADH carries electrons from that broken down glucose. So we're stripping out electrons and transporting them on. Okay, now we are about to enter the Krebs cycle, the citric acid cycle. Let me zoom out a little bit. We have our acetyl coenzyme A, it's going to go in here. And enter this multi-step cycle that is even more confusing than glycolysis. Like, there's so much happening here, and we just don't care about most of it. But there are certain things that we do care about. So those certain things we're going to focus on. I am much more concerned with just overall general processes and you understanding how they fit together. The citric acid cycle is all about stripping electrons and harvesting all of the energy out of the pyruvate. We're taking pyruvate and we are just sucking it dry. We're taking out all of the free energy from this thing. And we're doing that by removing electrons and transporting those electrons as NADH and FADH2. Which is just another electron transporting molecule. Okay, so what we're going to see now is we'll have some oxidation occur, and we generate some carbon dioxide. This is where all of that carbon dioxide, so the Krebs cycle, this is where the carbon dioxide is getting produced in our original equation. Here's that CO2, it's made from the Krebs cycle. Okay. And it's just by oxidizing this molecule. And every time we oxidize this thing, we are removing electrons from it. How do those electrons then get moved to the electron transport chain? NADH and FADH2. So we oxidize this acetyl coenzyme A. The electrons get removed. We release carbon dioxide, and then those electrons get attached to our FADH and the FADH2. Okay. And then those get transported to, guess where? The electron transport chain. We're just harvesting energy. Harvesting energy and delivering energy. All right. We get a little bit of ATP here, and the ATP that gets generated here is from substrate level phosphorylation, which is something else that's in chapter 9. It breaks down the 2 different types of phosphorylation. So this is done through substrate level. And we don't get very much here, but we get a little. We get 2 of them. And I forgot to put that back here on glycolysis. Glycolysis here is also from substrate level phosphorylation. And we get 2 more here. Now, overall, in perfect conditions, aerobic respiration will generate 38 ATPs. We've only generated 4. So the big payoff hasn't occurred yet. All we've been doing up to this point is just harvesting and storing electrons. So we're about to make the magic happen here in a second. Alright. And we're in the mitochondria right now. Okay, so there's some more stuff that I want to add about the citric acid cycle, but we're out of room. So I'm going to come down here, and we're going to add some stuff. let's do this. I'm going to add 2 major points. So once acetyl coenzyme A is formed, the Krebs cycle is all about oxidizing the pyruvate and generating NADH and FADH2. Okay, so once acetyl coenzyme A is formed, the Krebs cycle focuses on generating NADH and FADH2 for the electron transport chain. We are picking up free electrons, so we're going to go drop them off. So here's the main point for the Krebs cycle. Let's separate these. The Krebs cycle harvests all of the stored energy from the pyruvate. And it does so as NADH and FADH2. So we're bleeding this pyruvate dry. We're taking all of the free energy out of this thing by harvesting its electrons. Does this make sense so far? All right. So what do we have now? If we were to zoom out and just kind of look at where we're at, we have taken glucose, we have broken it down into pyruvate, generated a little bit of ATP along the way, but not much. Because we're going to have to spend some more ATP to get the pyruvate into the mitochondria anyway. Yes?
So is the little bit of ATP that's to keep the
Well, yeah, and it's hard to say, like, it's not like it's set aside and used for anything in particular, but if you were to just start saying where's ATP gained and lost, then essentially yes. Especially since we have to burn some to get this stuff into the mitochondria anyway. So now we get it into the mitochondria as pyruvate, and this is only in the presence of oxygen. So actually we should add that in the presence of oxygen. Because without oxygen, we're headed to finished, to a different pathway. Then the pyruvate is going to start being oxidized over and over and over again. We oxidize it at first, and we kick out this carbon dioxide. We oxidize it again, and we get some NADH. We oxidize it. We add some the coenzyme A, and that allows it to enter the Krebs cycle, where we oxidize and oxidize and oxidize in order to produce NADH and FADH2. We're just stripping out electrons. Okay. We generate a little bit more ATP, but again, not much, only 2, and now it's time for the big payoff. So all of these electrons are going to get dumped now into the electron transport chain, and that's where we're headed next. So I'm switching over to purple now. Again, I'm trying to keep this consistent in your book. Your book used blue, red, and purple for the same processes. Hopefully this will make it match up better. Okay. So, electron transport chain. We are entering on this arrow right here. And you can see here it says electrons. So we are just piling electrons through this. Your book did a really good job of showing what this mechanism kind of looked like. And I meant to have this pulled up. But the electron transport chain is made out of this series of proteins that are embedded in the membrane of the mitochondria. Here we go. That's what I was looking for. And what we're going to see happening is electrons are going to come in from the NADH and the FADH2, and they're going to fall down the series of cascades of proteins. And they're actually being pulled down these little stair steps, and the whole way down, they are releasing that stored energy. And that energy is going to be used to pump hydrogen ions outside of the mitochondrial membrane to set a concentration gradient. What is actually pulling these things down this series of steps, though? There's a particular atom that's doing this, or a molecule, that has a really high electronegativity. That's pulling these electrons down the electron transport chain. Oxygen. That's the role of oxygen. The reason why oxygen is needed, glucose plus oxygen. Oxygen is the final electron acceptor. It is pulling the electrons down the electron transport chain. And as the electrons make it to the oxygen, what molecule is made? Water. There's the water. That's why water is a byproduct. Okay? So oxygen plays this major role, and this is oxygen's only role. So the reason why aerobic respiration uses oxygen is because oxygen works as that final electron acceptor to pull electrons through the electron transport chain. Okay. So I wanted to highlight that so that whenever I went in over this, it kind of makes sense. Okay, so electrons enter the electron transport chain. They're being pulled across that series of proteins. Okay? And as the electrons move through, they're getting pulled by this oxygen right here. And remember that oxygen has a really high electronegativity. Do this in purple. High electronegativity. And that's pulling those electrons down, and then it's going to end up forming water. And this is that water. That's that byproduct out of cellular respiration. So I just colored it kind of a darker blue, but you don't have to do that. Okay, so then we end up with water. All right. So let's actually write out some of these steps. So step 1, and that would be occurring right up here at this intersection. Okay. I'm going to use the E minus, that means electrons. So E minus means electrons. From, oh man, that's supposed to be an N. NADH and FADH2 are pulled through the ETC. Towards oxygen. Yeah, so that is the mechanism that's going to start releasing energy. Now, this energy that gets released, this releases free. That release of free energy is going to be used for our next step here. They serve the same major function, they're just structurally different. Yeah, they're big and complex, and I don't remember what their overall structure is, but they're nuts, they're huge, and it doesn't matter to us. But they're coenzymes, so they're a protein complex. Okay. Now, what is this doing? Well, as those hydrogens fall down that cascading series of proteins, what those proteins are is they're protein pumps, and they're hydrogen pumps. And the energy from those electrons falling through the proteins is going to start pumping hydrogen ions. So hydrogen ions are pumped across the mitochondrial membrane. This sets up a concentration of hydrogen ions, to where now there's going to be more hydrogen ions outside of the inner membrane of the mitochondria than inside. And that's what we see here. So we had hydrogen ions here, being actively transported by the electron transport chain to the other side of the mitochondrial membrane. Okay. Now, we have a higher concentration here than here. Which way do these hydrogen ions want to go? They're going to want to fall back across to a lower concentration, right? And this is where the energy is going to come to make ATP. Yes? In this case, it's using the flow of the electrons to do it. Okay. So now let's look. So step 2, that was happening here. We have the pumping of those hydrogen ions. Okay. Then we're going to look right here for step 3. And actually, I'm going to move us over here to oxidative phosphorylation. The last step here is oxidative phosphorylation. This is the big payoff. This is where we're going to start cranking out some real ATP. What's up? They're two different processes, but they're related but separate. The electron transport chain's job is to set up a hydrogen gradient, hydrogen ion gradient. Oxidative phosphorylation is now going to use that hydrogen gradient. Okay. So now in our third step here, the H plus gradient is used to power ATP synthase, which is the enzyme that takes ADP and phosphorus and tacks that third phosphate group on to make ATP. So this is going from ADP plus P to ATP. That requires energy, and the energy that is used to do this comes from this hydrogen ion gradient. Okay. Now it does this through a vocab word that's in chapter 9 called chemoosmosis. And this is the use of a concentration gradient to do cellular work, to provide energy to the cell. Chemoosmosis. CHEMIOSMOSIS. Chemoosmosis. And then our last step here is ATP is generated. Through oxidative phosphorylation. A bunch of it. Numbers vary, it depends on the cellular conditions, but anywhere from 32 to 34 ATP is what you'll typically see. This means that through the whole process, we've had anywhere between 36 and 38 overall. 32 to 34, sorry. My timestamp is over it. Okay. So those two steps are occurring here. We would have the hydrogen ions being pumped across the membrane. Now we have a higher concentration here and a lower concentration on the other side of the membrane. That's going to cause these hydrogen ions to fall through the ATP synthase protein, which is going to use that energy provided from the hydrogen ions falling through to tack that third phosphate group onto ADP. And that's where we end up with all of this energy right here. So bunches and bunches of ATP. That's why it's this bigger starburst. We're getting a lot more out of it. That's the big payoff. That's what makes aerobic respiration so much more efficient than anaerobic, is how much more ATP we're getting out of it.
How many ATP are processed?
Anywhere between 36 and 38, there's debate on that. Okay. What I'd like to do before we run out of time is just very quickly try to sum up this process into just like a couple statements, and I feel like that will make this easier to digest. Okay. So this is a 4-step process. The first thing that we're going to see is glycolysis. In glycolysis, the primary function is to take glucose to pyruvate. Now, along the way, we're going to make NADH, and we're going to make 2 ATP. But the primary function is to generate pyruvate. After glycolysis, that pyruvate is going to enter the Krebs cycle, or the citric acid cycle. In the citric acid cycle, our primary function is going to be to harvest electrons from pyruvate. Okay. Along the way, we end up making 2 ATP, but we do this through storing electrons in NADH and FADH2. And we generate 2 ATP. Okay, so we just removed a whole bunch of electrons from, storing electrons in NADH and FADH2. Run out of room on here. Then we move into the electron transport chain. In the electron transport chain, electrons are used to set up a hydrogen ion concentration gradient. Okay. And in doing so, we end up releasing water. Because remember that oxygen was used to pull the electrons through the electron transport chain, and that ended up generating that water molecule. Okay, and then final step, the big payoff. Oxidative phosphorylation. This is a vocab word out of your book, proton motor force, which is just the force generated by those protons falling back through the ATP synthase. It is used to drive chemiosmosis. No! Via oxidative phosphorylation.
Okay. Matthew Sadler, Macy Sadler,